And it's my pleasure to introduce Nisha Shrestha from Nepal. She completed her bachelor's degree in the field of aquaculture and fisheries, and following her passion for aquatic life and ecosystems. The next step in her academic journey, she went on to achieve a master's degree in aquaculture, environment, and society through the Erasmus Mundus program, which gave her the opportunity to study in several European countries, including France, Greece, and Scotland. She's currently a first-year PhD student in the Aquatic Animal Health Lab at Michigan State University, and she's going to present the start of her work here today. So thank you very much, Nisha.
Thanks a lot, Dr. Phelps, for introducing me. Hello, everyone. I'm Nisha. I'm extremely excited to present my work today that I started like 3 months ago. And I'd like to thank all the organizers for providing me this opportunity. This is my first presentation in any kind of professional meeting, and of course, my first talk in the fish health section summer seminar series. Today in this presentation, I will talk about the ongoing studies focusing on determining the genetic and serotypic diversity of Flavobacterium psychrophyllum in North Central region's salmonid facilities. I'd like to take a moment and thank all my co-authors for the contribution in this study. First of all, I want to introduce myself since I'm a new member of Fish Health section, and I look forward to meeting you all in person someday. I'm from Nepal. I did my bachelor's degree in aquaculture and fisheries from Agriculture and Forestry University in Nepal, where I was mostly involved in carp and tilapia breeding and aquaculture works. I wanted to continue my career in this sector, and fortunately, I got Erasmus Mundus scholarship to study Masters in Aquaculture, Environment and Society in Europe. So with that, I was provided an opportunity to study in different European countries to complete my master's degree. I studied mostly the shellfish, focused on mussels, oysters, clams, anatomy, biology, toxicity, and culture systems. I did my master's dissertation in Scotland, where I studied about relative abundance of harmful algal bloom using the meta-barcoding tool, which is a molecular way of species identification and study. So after that, I feel like I'm done with shellfish, done with harmful algal bloom. And then I started looking for studying about finfish and disease ecology side of it in the US. I reached out to Dr. Luck and joined Aquatic Animal Health Lab here in January this year. And I'm really excited for my journey in here. So I would like to highlight the status and importance of aquaculture globally at first. Aquaculture has experienced significant growth over the past few decades globally, and it's considered an important sector to meet world's increasing demand for protein. So it has been growing at a faster rate than the capture fisheries since the late 1990s, as you can see in the graph. But in the US, large part of the production is lost due to disease. For an example, in this chart, the orange and the blue bar as a total represents the total loss of trout intended for sale, and the orange bar represents the loss of trout due to disease. So looking into the loss since 2005, about 51% to 91% of the total trout intended for sale is lost due to disease, and the flavobacterial disease is considered as one of the top contributors. The Flavobacterium psychrophyllum, it's a gram-negative, rod-shaped, golden yellow pigmented bacterium belonging to the class Flavobacteriaceae that causes rainbow trout fry syndrome and bacterial cold water disease, responsible for a significant loss of cold water fish globally. Flavobacterium psychrophyllum was initially described and recovered in 1948 in coho salmon, and now it's problematic in many states in the US. And in severe cases, if you can see, this bacterium causes severe necrosis of the caudal peduncle region, eroding the muscles all the way down to the bone, exposing the vertebral column, as you can see in this picture. And if this disease goes untreated, it can cause losses greater than 70% in infected rearing unit, which is why this disease can be the significant impediment in aquaculture facilities and also in the hatcheries that raises trout and salmon for conservation purposes. So why this bacterium is so problematic? It's because it can be transmitted horizontally, meaning from infected fish to uninfected fish. And another problem is that this bacterium can be efficiently transmitted vertically, that is from parents to offsprings through the infected reproductive fluid. And there is also the reduced antibiotic susceptibility to the US approved antibiotics, and there is no any commercial vaccine available against this pathogen in the US. Bacterial cold water disease is also the most concerning disease in North Central region, the region which is the target region for our study. This project is regional, aiming to benefit the NCR producers. Lots of travels and lots of visits to farms to interact with producers are also the parts of this project. Dr. Kavis is leading these and all other large aspects of the projects. So with his communication and interaction with the farmers, it's known that the numerous farmers and hatcheries are experiencing increased mortality rates and minimal success in hatching among their trouts and salmon. And these losses can be attributed to direct or indirect consequences of Flebobacterium saccharophilum. Though this disease is so concerning, there is lack of state-specific saccharophilum study in North Central region. Well, this huge diversity of Flebobacterium saccharophilum seems to be likely a factor for inconsistencies of preventive and control measures for this disease. You probably have seen this eBreast diagram several times during the presentations by our lab. There's a lot going in this, but the important takeaway from this is that the diversity of variants of Flebobacterium saccharophilum is huge, and every number in here represents particular phenotypic variant of saccharophilum, and this highlighted with red represents the genotypes, those discovered in the US, and in many variants in the US that are distinct from the variants found in the other countries. And these and other studies generated the information that was shown in the previous slide. So this map is from one of those studies that represents the current information on the number of saccharophilum isolates typed from different US states. And only 5 states among the 12 states in here, in the North Central region, which is blue in here, are covered for saccharophilum genotypic diversity study. It also has to be noted that those analyzed isolates originated from the state and federal trout and salmon facilities, but the saccharophilum variants driving losses in private trout and private salmon farms are largely unknown. The genetic diversity of fish does not necessarily reflect their immune response. The serovariation, which refers to the variation in surface protein, can actually influence how the fish immune system recognizes the pathogen. Therefore, it's crucial to understand the serotypic diversity as well. In 2017, Roche developed a standardized method of determining the serotypes that could be used consistently across the laboratories. This method helped to determine which serotypes previously determined by ELISA and slide agglutination. As you can see, those mentioned here, like FT, FT, FPT, TH, FPD, and so to the newly molecular serotypes 0, 1, 2, and 3. And in 2020, Herrera identified type 4 serotypes originating from type 0 and now giving us 5 different serotypes. The pie chart represents the serotypic diversity of Flebobacterium saccharophilum in the US. This is a work of a PhD student in our lab, Sean Lennox, who has been elucidating the serotypes of the US isolates, which were largely unknown before. Though type 0 looks common in here, it should be noted that type 0 is a catch-all of some unknown molecular serotypes. I'm not going to explain in too much detail on this, but I'd want to highlight that it's important to have understanding on the serotypic diversity too, to decide on which isolate to prioritize for vaccine development work. I'd also want to highlight that the most of the isolates in these studies were not from NCR. There are several great approaches in vaccine development, including live attenuated and bacterians done for studying the efficacy of monovalent and polyvalent immersion and injection vaccines, showing them to be very efficient. But these studies possibly might have only been effective to the region they studied or to the Flavobacterium microphilum strains they targeted. Two studies done in the UK, University of Stirling, clearly shows the promising results of polyvalent immersion and injection vaccine in rainbow trout. The important message from this is that they characterized geno and serodiversity and made bacterian preparations for their region's dominant variants. We are going to do the similar approach in North Central region as well. So the hypothesis of my study is characterizing regionally dominating geno and serovariants of Flavobacterium microphilum in North Central region will inform the development of efficacious bacterians for bacterial cold water disease prevention. So to address this hypothesis, the first objective of my ongoing study is to determine the predominant variants driving the economic losses in NCR trout and salmon industry. Now, I'm going to explain in brief about the overview of the methods. On-site visits to the farms in each NCR states are done. The samples are collected, maximum 20 fish, depending on the size of fish, and shipped overnight or driven to Michigan State University Aquatic Animal Health Lab. And based on the conditions and size of the fish, it's decided either to ship them alive or ship them dead on ice. And after that, the clinical examination is done. The bacteriological exam is carried out on all fish. The cultures from different organs are taken for bacterial isolation in FPMA media. I will talk about the organs for the culture after some time. And the yellow pigmented bacterial growth are subcultured on the first media and inoculated in broth, and they are cryopreserved at -80 degrees Celsius for future identification and analysis. And the yellow pigmented bacteria are analyzed using the Sacrophyllum specific endpoint PCR assay by Toyama 1994. And the resultant PCR products are electrophoresed and then visualized under UV transillumination to confirm identification of Flavobacterium microphilum. And then the bacterial isolates confirmed as Sacrophyllum are genotyped using multi-locus sequence typing. Basically, multi-locus sequence typing, or MLST, is amplifying the 7 housekeeping genes by its specific PCR assay and then sequencing those amplicons, which allocates the isolates with their sequence types. And they will be molecularly serotyped using Flavobacterium microphilum specific multi-PCR assay, which assigns the isolates to their respective serotypes. My second objective for this study is to develop and evaluate the efficacy of bacterian preparations in preventing the losses caused by the predominant Sacrophyllum under the lab and field conditions. So the predominant Flavobacterium microphilum variants will be selected for the preparations of formalin inactivated bacteria to use in range of bacterian preparations. The monovalent and polyvalent bacterian preparations will be done for efficacy experiments. So with these all preparations, we aim to be able to produce the bacterium that can be deployed on each farm in the state. Now, I'm very excited to share some of the initial results that I have since starting the study a few months ago. We have sampled fish from 6 facilities in total: South Dakota, Minnesota, Iowa, Wisconsin, Michigan, and Ohio. You might be wondering who might travel thousands of miles just to collect the fish. Well, he is an invaluable collaborator and study co-author of our study, Dr. Kavis, who has been helping producers with so many things to improve fish health, and also has been leading the fish sampling collection and shipping back to us. So the total fish from all the facilities was 117 trout and salmon examined so far. Next, I would like to share some of the gross disease signs we observed in the moribund fish that we targeted and collected in this study. Disease signs that we observed included different degrees of fin erosion, severe ulceration at caudal fin and caudal peduncle, gill pallor, exophthalmia, and anophthalmia, varying degrees of dermal ulcerations, and skeletal deformity. Other than this, we also observed general fraying of fins and abdominal distension. The gross internal findings include paler internal organs, which is shown in the figure, including pale kidney, pale liver, pale heart, and the proliferating ulcers damaging the viscera. So in some cases, the ulcer I showed in the previous slide can penetrate all the way through the body wall and damage the internal organs, as shown in the picture in right here. So 456 primary cultures were taken from gills, brain, spleen, kidney, dermal ulcers, fin erosion, and eyes from 117 fish examined so far. From those cultures, 171 subcultures of yellow pigmented bacteria were taken, and nucleic acid extraction was done using DNAZ blood and tissue kit, after which the Flavobacterium psychrophyllum endpoint PCR assay confirmed Flavobacterium psychrophyllum isolates from them. And among them, 118 isolates were confirmed as Flavobacterium psychrophyllum, which was from 50 out of 117 fish with about 43% prevalence. And 80 representative Flavobacterium psychrophyllum isolates out of 118 are selected for multi-locus sequence typing. Out of those 67 isolates have been completed so far, and the sequence results are coming in soon. And I cannot wait to begin analyzing them and finding what I got until now. So far, the state sampled, we recovered Flavobacterium psychrophyllum from fish from 5 different states, 7 by 22 fish, which is 30.4 from South Dakota facility, 12 by 20, which is 60% fish from Iowa facility, 8 by 14, which is 57.1, 19 by 20 fish, which is 95% from Michigan facility, 4 by 20 fish, which is 20% from Ohio facility. We did not recover any Flavobacterium psychrophyllum isolates from the samples in Minnesota facility. The fish from there were very small, about 0.1 to 0.3 grams, due to which culture of most of the targeted organs were difficult. We got many other yellow pigmented bacteria that in some cases could have outcompeted Flavobacterium psychrophyllum, though not known. We likely will sample there again in future to investigate. To conclude, bacterial cold water disease is widespread in NCR state facilities we sampled. So investigating the variants driving losses within the NCR trout and salmon facilities is crucial to understand the preventive measures that we can use to reduce the loss. This study has already analyzed the samples from 6 state facilities, 2 states, Ohio and Iowa, where the Flavorbacterium saccharophilum diversity study was not done before. And our goal is to sample facilities in many of these states as possible. And after that, we will work on producing autogenous bacterium that can be deployed in its farm in NCR states to help farmers prevent bacterial cold water disease in their facility. The next steps would be analyzing more fish, genotyping and serotyping the Flavorbacterium saccharophilum isolates recovered from them, preparing autogenous bacteriums, and evaluating its efficacy in a lab and farm level. Yeah. So finally, I'd like to acknowledge USDA NIFA and ICRA grant for funding this research, all the collaborators for teaming up, including Animal Health Lab members for their help and support, Dr. Myron Cabus and my advisor, Dr. Thomas Luck, for guidance, expertise, mentorship, and many more. Thanks everyone for tuning in. And with that, I'm happy to take questions.
Excellent job, Nisha. That was a very good presentation and exciting early results.
Yeah.
Folks, if you want to drop a question into the chat box, feel free to do so, or you can unmute yourself and ask your question directly.
Hey, Nisha, I'll jump in. Fantastic talk. Great job. You guys continue to do fantastic work up there. I'm just curious, do you have a cutoff? You know, you can always get more isolates. There's always more fish. Is there a cutoff for when you're going to stop sampling and say, all right, my data set's big enough, we're going to use this and move on to the next step?
So thanks for the question at first. That was a great question. So we are planning to get at least one facility from the 12 NCR states, but we do not have any cutoffs. So I think as far as we are good with 12 NCR states fish, yeah, we'll plan on getting on further steps, but we do not have cutoff for now. So we are planning to get more and more. So we are on that side for now. I hope that makes sense.
Yeah, no, absolutely. Yeah, it's just easy to get that, you know, isolate creep. So at some point, yeah, you're going to want to just say, all right, enough's enough and move on to the next steps. But great talk. I'm excited to see how this develops.
Thank you.
We have a couple questions here in the chat, Nisha. The first one's asking about, can you explain how the PCR bands are used to identify the isolates and how that approach can help distinguish different strains used for the bacterium development?
Is that the question? The PCR for different isolates means specific isolates of Flavorbacterium saccharophilum?
Yeah, so if I understood it, the PCR bands were to identify whether or not it was saccharophilum. And then you'll have a second test to determine the different strains.
Okay, so we are following Toyama principles for doing PCR, and then we have the PCR reactions. We have the positive isolates, and then after doing PCR, then we get a band of similar size, and from that we can confirm whether or not the isolate is Flavorbacterium saccharophilum or not. And after that, we use multi-locus sequence typing principle, which is, as I mentioned before, amplification of the 7 genes of the isolate. And then from that, we will get the bands of 7 genes, which after looking the chromatograms, we will find if it's okay or not. And then we will send that for the sequencing. And after getting the sequence results, we will have to plug into the specific software. And then after that, the different genotypes of the Flavorbacterium saccharophilum will be known. So after that, we will identify which isolate it is. I think that makes sense as well.
Yeah, thank you. And then another question, why do you think the prevalence was so high in Michigan compared to other states?
So the facility we went to Michigan was, the most of the fish. So I and Dr. Locke and the team, we all were there in the Michigan facility. And then through our eyes, when we looked at the fish, were very lethargic and looked sick. So I do not have like specific answer to answer you why in Michigan it's very prevalent, but I can say that it's prevalent in Michigan facility that we sample.
Okay. And Ben, I see your hand up.
Yeah, great talk, Nisha. I had a quick question. Maybe I missed this at the beginning, but are all the facilities being examined? Are these all commercial aquaculture? Are they perhaps a mix of like state DNR facilities as well? And just along that lines, I was curious if you knew the source of those fish, if they were all from commercial, if you were kind of tracking where those fish came from, as there may be a common source of a different sequence type associated with the source of the fish.
Okay, so thanks a lot for the question. That was a good question. And I was expecting that would come today. Yeah, for your first question, either if not, the fish came from commercial farms alone or also from other public hatcheries and all. Yeah, so the initial idea was to sample only the commercial farms, as we are concerned about improving or preventing the bacterial cold water disease in NCR states commercial farms. But then after that, we realized and we know that there are commercial farms only in 4 states out of the 12 states. So now we are planning to do, to collect the fish and sample 4 of the commercial states. And we are also taking some of other, the public hatcheries or private hatcheries as well. But I cannot tell you the name, like from which one we sampled, but it's of course the mix of public hatcheries, public facilities, and the commercial farms. And sorry, what was the second question? Can I get it again?
I was just curious for the commercial farms,
Mm-hmm.
if Dr. Cabus is keeping track of perhaps whether they got eggs from a commercial source, that perhaps you may find a particular sequence type associated with the source of the fish.
I really do not have idea about this, like from where they got, but the farm where I was there along with the team, they got fish egg from the commercial vendors who produce genetically bacterial cold water disease resistant strain. So I'm sorry, I do not have really great picture of from where they got, but yeah.
Great, thanks. Another question here in the chat. Do you know from the yellow pigmented bacteria that were cultured in Minnesota what species they were?
So talking about the morphology of the yellow pigmented bacteria, that was gliding and covering all the plate in like 1 day of incubation. And then there was a kind of omelette kind of looking, the yellow and then surrounding undulated kind of morphology found in most of the plates. So we sequenced that and we only got Flavobacterium genus confirmed, but we do not know more than that, as the project's target is just looking to Flavobacterium psychrophyllum. We didn't bother in getting too much in detail on this, but it's on our plan that someday we might want to go back and then look which species it is. I hope that answers the question.
Yeah, thank you. And one last question from Haitham.
Great job, Nisha. Excellent talk. It's a kind of follow-up question to what Matt asked originally in the beginning. And again, I might have missed it because I joined 15 minutes late. So what are the numbers, again, for the isolates you collected versus the fish that you sampled? Because I think that you have more isolates than the number of fish, right?
Yes.
That means you have more isolates, more than one isolate from a single fish.
Yes.
So now you classify those as isolates, not strains. What are the chances that you have duplicates? You have the same strain again, because you have maybe 2 or 3 isolates from the same fish.
Yeah.
And you know, did you have a cut off for how many isolates you get per fish?
Thanks a lot for the question. Yeah, as per the slide, we collected different organs, the bacteria from different organs, and then it might be possible there would be the duplicates of the isolates. So after doing, but while selecting the isolates for doing MLST, we prioritized the different organs, like, so for an example, I got isolates from gills, brain, spleen, and kidney, all organs from one fish. So we were trying to select the one that is the representative of most of the overall study that we want to do. So yeah, there might be duplicates, but after doing genotyping, we will know which sequence type it is, and then from there we can think about what to do further. So for now, we are doing as much as possible, or as much as trying to collect more isolates and trying to do more overall study. Yeah, just not to miss any. Thank you.
Yeah, excellent work. Good job.
All right, thank you. Nisha, I just put your email into the chat box. There's a few more questions that folks have, but for time, we're going to stop here.
Okay, thanks a lot. And you can reach me out for any questions. I'll leave my email in the chat box.